we'll go ahead and continue with our presentations. My name is Tony Gondola. I'm the physiology instructor for FSI. And our next presenters are colleague Ratekin and Sofia Simina. And they can present the work they did looking at snake venom and their effect on cancer cells. So.
Hi, so I'm colleague Ratekin, and my name is Sofia Simina. We investigated the effects of anti-neoplastic qualities that are associated with the Crotalus Aquilus snake venom on both human colon cancer as well as human lung cancer. So a brief introduction into cancer itself. We chose to study cancer because it is the first leading cause of death among people under 85 in the United States, and the second leading cause of death overall. Also, lung cancer is the second leading cause of death as far as cancer goes, and colon cancer is the third leading cause of death. Those are the two cancers that are specifically tested. Cancer works because, and it hurts you because it just continually divides. Instead of stopping and staying in G1, which is up there, and doing its job, it goes straight to these checkpoints, which are the red things, which normally tell the cell to stop and do its job and not divide. But in these cancer cells, they just keep going and going and going and spreading. And when this happens, they don't do their job. It takes too much energy from the body to cause this problem. Also, these are colon cells, and the dark purple that you can see are the nuclei of the colon cells, and they're really irregular because, again, it's dividing so quickly. Okay, so a brief introduction on snake venom. Snake venom is a complex mix of proteins, enzymes, as well as polypeptides. There are two types of toxicity that is associated with snake venom. One type of toxicity is neurotoxicity, and it's associated with a lapid. An example of an elapid would be a cobra. And neurotoxicity affects the neuro section of a body, causing respiratory failure and ultimately death a lot of the time. And the second type of toxicity is chemotoxicity, and that's associated with vipers. An example of a viper would be a rouse. Chemotoxicity causes extreme hemorrhagic effects all throughout the body. So if there's a snake bite, it would cause hemorrhaging of the vessels around that snake bite. Some specific enzymes associated with snake venom would be metalloproteinase, and it's also known as SVMP. And also there's L-amino acid oxidase and disintegrin. Those are a few of the plethora seen in snake venom. Some FDA-approved medication that has been released onto the market would be Ancrode, as well as Ratekin. Ancrode and Ratekin have serine protease in them, and that is also derived from snake venom. Ancrode and Ratekin also help in patients suffering from myocardial infarction, as well as deep vein thrombosis and stroke. Okay, I'm sorry. So this is a picture of snake venom actually being extracted from the snake. In our lab, we spin it down and dehydrate it, and that's the snake venom that we personally work with, and a lot of other researchers do that as well. So we tested this all with Crotalus Aquilus Venom, and the common name for Crotalus Aquilus is the Crotalus rattlesnake, and it's found right up here in the plateaus of Mexico. It's relatively high up, and it is found in big rock piles or very thin vegetation. It likes to hide out a little bit. There are some studies going on to see how the increase in agricultural land in this area is affecting them and how their habitat depletion is affecting them. Also, they eat small critters like crickets and lizards, and they're flexing, so they use their venom to ingest their prey and not just for digestion. They're relatively small, about 20 inches in length, and stout bodies. They go anywhere from brown and gray to green in color. Okay, so you may be wondering what our purpose was. Our purpose for our investigation was to look at the properties associated with the Crotalus Aquilus Venom and see if it had any anti-metastasis effect on the colon cancer as well as on the lung cancer. The specific
that we used was Colo 205 and Lung A549. The Crotalus Aquilus Venom that we worked with has not yet been tested on those cancer cell lines. That's why we chose those, as well as the facts that Carla gave you previously. So a little bit about how we tested this venom in its toxicity towards the cells. We used what's called MTC assays, and that's seen up here. The darker purple is where there are more cells, and the lighter there are least amount of cells. And it goes in, and the reagent stains the mitochondria of the cells, and it tests their metabolic activity, which in turn gives us a good estimate of how many cells there are. After we do that, we put in a detergent, which lyses the cells so that we can put it into a plate reader and get a lighter fluorescence. We also use an HPLC sized solution, which is where we took the crude venom and separated it, and we got 7 sheets. We separated it in 7 parts, each by size, and they're identified by their light absorbency. So this lets us test just a certain part of the venom to see what exactly is going on. We also did gel electrophoresis to see what's in the venom. We did this for both crude venom and our sheets. And this gives us relative sizes of the different proteins and molecules in our venom so that we can kind of identify what they are. We found a lot of the proteases. There are usually mycotoxin and disintegrants in venom as well. Finally, we did our own cell culturing. So we had flasks of adhesive cells, which means that they just stick to the bottom of the flask. So in order to get them out when we actually needed to use them, we used trypsinization, and that gets the cells out so that we can use them in our wells and test them. Okay, so this is a gel electrophoresis picture of the crude venom that we looked at. And it shows 3 specific individuals from the Crotalus Aquilus species, individual 34, individual 37, and individual 38. In rows, it shows the proteins associated with that individual, starting with P3, metalloprotease, and then going on to P1, hemorrhagic metalloprotease, as well as phospholipase A, which is a neurotoxin, and it is harmful to cell membranes.
And something to point out as well is that there is a bit of a difference between each of these, and that's because venom is very, very complex, and it can change in individuals from the same species, and it can even change within one individual throughout its entire lifetime. So it really depends on which snake you're testing. And in order to kind of get more accurate results, we pooled our venom, so we took them from several different individuals.
Okay, so this is the result for the Lung A549 MTC assay. As you can see, it's the venom concentration effects on the number of living cells in the MTC assay. On the y-axis, we have the number of living cells, and it's measured in cells per milliliter, and on the x-axis, we have venom concentration in milligrams per milliliter. The general trend of this graph shows a decrease. At 40 milligrams per milliliter of venom, we had 50,000 cells. When we got to the highest concentration, 99.89% of the cells were dead, which means that the venom did a really great job at actually killing what was in the well. The IC50 number that we recorded for this particular graph was 10.47 milligrams per milliliter of venom, and the IC50 number is the number of the amount of venom it takes to actually kill 50% of the cells. Right here, you can see that the control well is actually slightly lower in concentration of cells than the 0.3 well. This is because MTC assay actually measures the metabolic activity of the cells as opposed to measuring the amount of living cells versus the amount of dead cells. So when the cells in the 0.3 well were put under stress, their metabolic activity increased.
And you can see that same trend here with the colon 205. Again, on the y-axis, we have the number of living cells, and on the x-axis, we have venom concentration. There's that same trend that as the venom increases, the amount of cells decreases. This cell line was a little bit harder to kill with the venom. Our IC50 number, or the amount of venom that it took to kill half the cells, was 13.86, which is a little larger, obviously, than the 10 that was found with the lung cells. Also, 98.9% of the cells died at the highest concentration.
right-hand side shows the HPLC side exclusion chromatography. The peak that we focused on is peak 1 with the star right above it. This peak is the peak that had the highest amount of light absorbency, which is shown on the y-axis, measured in AU, and the time is shown on the x-axis, measured in minutes. This is the peak. All 7 of these peaks were then tested in a gel electrophoresis that's shown on the right. Right over here, you can see the ladder, which is the known substances that we compare the peaks to on the right-hand side. So right here is the second column, and this shows that the first peak that we looked at previously has T3 metalloprotease in it. And in the other sections, there's also P1 metalloprotease and phospholipase A2. We did do an MTP assay on that first peak. However, our results were inconclusive, so in the future that should be tested again. And as far as error goes as well, we tested everything in triplicate. So on each 96 well plates we tested for the MTP assay, we did it 3 times, and we each did one. So each result that you see has been done 6 times to try and get out a lot of the error. We overall found that the venom from Crotalus Aquilus snake does kill both lung and colon cells. We also found that the venom is very, very complex. We saw over 30 bands on our gel electrophoresis, and there are actually more compounds in there that just happen to run at the same speed, so they look like one band. In the future, we need to try and separate these so we can see exactly what's killing our cells, and again, from that first peak, and then separate it further. All right, so as Carly pointed out, the venom itself, as in the crude form, did have anti-cancer effects in it because it had anti-cancer agents. The fractionized venom could have a potential effect. That's why further research needs to be done, and those do need to be further tested. After those peaks would be tested, the single enzyme should be fractioned out using iron exchange chromatography potentially to see if those peaks could have pharmacological uses and should be further developed into medication as described before as antidotes. We would like to thank the SL Energy Foundation as well as the Clinton Family Foundation for not only sponsoring our research, but for also sponsoring our stay here at FSL. We'd like to thank Tony Gondara and Anthony Saviola for helping us in the lab and with our research that we did. We would like to thank Dr. Steve Maxey, Dr. Archie Musajuri, Dr. Patrick Burns, and Brittany Law. They were all working in the lab and were very helpful throughout this whole process. We'd like to thank the McAfee Lab for helping us and for letting us use the space. We'd also like to thank the University of Northern Colorado for housing our space here. And we'd also like to thank the Frontiers of Science Institute for this opportunity. Now we'd like to open up the floor to any questions that you could. I'm not exactly sure on the length of time that it would take for the drug companies, but I know it does have to pass an extensive process to become FDA approved because they have such high standards on what they release to the market, and a lot of the times they will do longitudinal studies in order to see the effects of the drug throughout a certain period of time. So they would have to test the drug and perhaps wait, up to 5 to 10 years potentially to see the side effects of the drug, and so that they have a small sample size if anything was to go wrong with the drug and that person. Charlie?
What is the actual process for going about collecting venom from the Crotalus?
Okay, the question was, what is the actual process that is used to go about collecting venom from snakes in general?
It's
relatively hard to find. Ours was collected a few years ago by two individuals near Mexico City. And it was seen up there at the beginning. And the venom extraction, you take a cup with a membrane over it, and you hold the head, and you milk the snake for front snake. It's a little bit different for rear fang snakes. I know you have to put them to sleep and such. I don't know the exact process, however.
So this is a picture of how they extract the venom from the snake. And they hold the snake by the back of its neck right here. And they put the thing in the cup, and then the venom just seeps it.
And that's actually done here at the University of Northern Colorado. It's something that we have several snakes in the basement here.
More than several. We have a lot of snakes in the basement. All right, so are there any other questions?
How does one ensure that the venom not kills the somatic alone and anti-neoplastic?
Okay, so the question was, how does the venom not kill the somatic cells or the normal cells and target just the harmful cells? So basically by using crude venom, you have venom that's composited with a plethora of enzymes and proteins that are associated with it. By using ion exchange chromatography to isolate just single enzymes, those are the enzymes that you would focus on for causing apoptosis and necrosis in the cells that need to be killed, as opposed to all the cells in the culture.
And some studies have been done in vivo with rats, and they injected disintegrants, which they target certain integrins in cells, which shuts off the metastasis and growth. And that was done with mice or rats, mice. And they actually found very little side effects in other cells.
Aisha? I can't hear your question.
I got it. If it targets certain cells, how do you make it target the certain cells? How do you keep it localized?
So obviously certain cells have receptors and certain enzymes will bond to those receptors. So by isolating the enzyme, you isolate the enzyme with the receptor.
So it depends on the particular kind of cancer one enzyme. Sure. Yeah, and very, very particular.
And as you saw from the presentation, some venoms are more potent towards some cancers as opposed to others.
So do you think that the crude venom from Crotalus Aquilus would work on another type of cancer, like maybe lymphoma or leukemia, or is it specifically for the lung cancer?
Okay, so the question was, would crude venom work on a different sort of cancer, or is it specific to colon and lung cancer?
And absolutely. Crude venom will kill most cells because it's very toxic. It has been studied, I know for sure here, on breast cancer cells, and it works very well.
So I know that for small concentrations of venom, you actually saw what appeared to be an increased number of cells because of increased cellular activity in the grass. Are there any ways to avoid that?
Okay, so the question was, are there any ways to avoid the stressing of the cells? I'm